So I'm going to speak about reproducing catalytic micelles as early nanoscopic protocells. And this is a result of about 25 years of work that have to do with the GARD model that I will speak about with some detail. The harbouring dynamic lipid aptamers in my title is just a small part of what I'm going to speak about. I'm going to start with something unusual, the human sensory homunculus. And this is a lesson regarding relative importance. So when you look at sensory, you see that the hands and the tongue have a very large part of the story. What about LUCA and evolution and origin of life? The popular view is that there is a small trunk at the bottom and then the rest is evolution. But in fact, it looks as if I should speak about the origin of evolutionculus, which means that the crux of life evolution is a very long, a large trunk before LUCA. Not that people don't agree to that, but most of the studies assume that you get to LUCA quite quickly, as shown, for example, here. There is non-living chemistries, RNA, metabolism network, maybe in an ocean, membranes that form spontaneously and so on. But what happens between that and LUCA remains a mystery. So here is what I'm going to, how do I get rid of this? Okay, yes, that's much better. Okay, the story is as follows. Our alternative look is that there has been a very long evolutionary process of pre-LUCA reproducing protocells. It's important, the word reproducing is extremely important. That is, it's not just chemistry roaming around. There must be entities capable of evolution that lead all the way from a very small entity, such as a one RNA or one lipid micelle or something like that, all the way to LUCA. And some of the details are graded evolution of chemically diverse reproducing lipid micelles harboring catalytic networks. So what we say is that reproduction started very, very early. And you can see that I show this indicating this, everybody here is capable of reproduction, just like we assume LUCA was. Now, what I'm going to speak about is beginning with a metabolism-like mutual catalytic network, then polypeptides may appear inside it catalytically, same for oligonucleotides, same of moving from micelle, which is very small, to a vesicle, which is larger and with a larger arena for chemistry, all the way to LUCA. Now, the idea that you can start not with lipid as a box, as a container, but lipid as an arena for chemistry dates back, among others, to Günter Blobel, Nobel laureate, who suggested that in very early life, functional molecules were located on the outside of membrane assemblies, and that it became a wall that harbors molecules inside much later. But there are many questions to answer. I'm telling you what I think, but I have to show how much evidence there is for that. Let's begin by the question, are lipids prebiotically available? Just two talks ago, we actually heard from Trigo or Rodriguez, we saw some of the materials that were produced by formaldehyde with a chondritic meteorite, and among them were lipids. But that's not just one story. Another story is Dave Deemer's very interesting paper showing quite a long time ago, 35 years ago, that when you extract molecules from a chondritic meteorite, you get lipid vesicles. More recently, we have collaborated with Philip Schmidt-Koplin on looking at mass spectrometry analysis of the Murchison meteorite. He did a very comprehensive analysis, and then we analyzed it together. And there is a very strong dominance of molecules that in mass spectrometry show a formula CnO2 with N equals 10 to 30. This is typically a trace or a fingerprint of a lipid. Or now, importantly, there is also an analysis that we wrote together in astrobiology 2 years ago, which has to do with the absolutely wonderful findings on Enceladus, the moon of Saturn. And this is a Postberg et al. And the idea is that there is an ocean, subglacial ocean, and there is abundant complex organics that when looking at other literatures, you can see that from the same kind of organics, you can get a lot of lipids. How about how would lipid gain the diversity that is necessary for what I have spoken about? That is that you can begin life with a mere lipid micelle. It cannot be just fatty acids, not just molecules that we call lipids today. They should be much more diverse. And there are examples in life today, such as you see here in the bacterial nucleolipid and in the bacterial lipopeptide. There are quite a few such examples. This means that lipids are not necessarily what we usually think about them as wall-forming, but they have functionalities that can be covalently bound to them. This is also including glycolipids and so on. I will not go into details right now. And in a large, wonderful database, lipid maps, there are, for mammalians, there are 30,000 different lipids. So lipids can be extremely diverse. Now, with this diversity, they can do a lot of stuff that lipids today cannot do. Some of these are just experiments that were done not in vivo, but some others are. In general, there are catalytic capacities for the smallest assembly of lipids, which is a micelle, and a lot of papers coming from the industry showing, for example, here, catalyzed attachment of a nucleotide to a lipid, azide cycloaddition, that's just a chemical reaction among many, catalyzed by proline derivatized lipid. So one of the biological amino acids participates here as a catalyst, a very simple but efficient catalyst. It is much more efficient than isolated proline because it's on the surface of a micelle. And this allows much more efficacy in terms of the substrates coming together, adhering to the surface of the micelle, and then undergoing the catalytic phenomenon. And the last one just shown here as an example, many others, there are lipid catalyzed tripeptide synthesis. So this goes in the direction of life as we know it today. But before I go on, let me say just one sentence. Life did not necessarily begin with with molecules of life as we know them today. Most papers assume that is the case. Most likely, that is not the case. I don't have time to say why that is so, but clearly, I'll say that at the last slide to some degree. Can lipids form so they can catalyze? How about forming a catalytic network, which is extremely important, as we learned from Kaufman and others? That it's quite clear that it is possible. You see here a paper by DeVarge from UCSD, and on the left you see here what he shows, and this is a network, and we even analyze this under the formality of what is known as collective autocatalytic sets, or RAF, and it is an RAF. So there is a catalytic network within an experimental system in which you can synthesize lipids catalytically, so lipids help catalyze their own formation. Let me skip this for a moment. How can fluid lipid assembly do any kind of recognition as necessary for beginning life? The answer is, and this is something that we've been working on in the last few months, the concept of dynamic aptamers. While the lipids are most of the time very fluid and moving around, by what we call mutually induced fit, a protein can localize or bring together certain lipids, and then the lipids will bind to the protein just as if a ligand binds to a receptor. There is a lot of literature on that too. Can lipid reproduce catalytically? The answer is not lipids, but assemblies of lipids. And this is a very famous paper by Louisi and others that is the picture showing here is that when you take an anhydride and you put it in water, it very slowly begins to hydrolyze. But once the fatty acid is formed, it forms micelles, and the micelles, after what I have already mentioned, catalyze much faster the hydrolysis. So you end up with a jump, a phase transition. Suddenly, a lot of micelles appear. And there is very strong evidence that what happens is that the micelles grow upon absorbing newly synthesized lipid and then fission so that actually they multiply. But can information be transferred from one generation to another in this kind of very simple reproduction device? The answer is that if everybody is green, if all the lipids are the same, there isn't really any information in that. But if you use a heterogeneous or mixed micelle, as shown in the drawing on the right, then there is a chance of transmitting information from one generation to another, but this is provided that the composition of the original small lipid upon growth will keep its composition as is, and then split and form two progeny that are very similar to the original ungrown mother. This kind of process can happen, but there is a very strong requirement here, and that is that growth will be homeostatic, concentration preserving. This is not trivial. Most mixed micelles will not do that. But we used a computer modeling device, which is the GUARD model, that can be simulated on the computer by what is known as Monte Carlo simulations. This is a rather complex slide, but let me go through it very quickly. On the left, we see a formula. This can be fed into the computer and find the fate, the chemical fate, under, these are kinetic equations. You can say what happens every minute and every second. And what we see is that under certain conditions, with a beta matrix, which is how much each of the resident lipid inside the micelle help come in some new lipids from the outside. If you have all of these numbers, and many of these numbers are actually based on real nature numerology, I should say, what we know from papers about reactions among molecules, when you do that, you find that on occasion, the lipid micelle will undergo this process with keeping its concentration preserved. This is the pink on the bottom right, and that is a multidimensional analysis of composition. And you see if it stays in the same red environment, it means that the composition doesn't change, which means that the progeny is like the mother. This is reproduction. Although we don't use here a sequence-based information, but compositional information, but I don't have time today to tell you about that. But most cells today, in most of what they do, use compositional information, such as proteomics, transcriptomics, and so on. So that's not new. Now, importantly, the composomes, as we call them, those positions in compositional space that breed through, those composomes are not only doing that, but they are dynamic attractors. That is, that life's probability is high. And that is because on a fast track, there is a fast track from randomity of micelle generation to those positions in which reproduction becomes possible. We also use molecular dynamics. No time to say too much about that, but this is an even better computational device to know exactly atom by atom what happens in a test tube. And there is a very strong belief today among scientists that what molecular dynamics shows is very, very faithful to what chemistry is really like. So we have two different lipids, and we asked whether the composition affects the rate of entry and exit. And what we found is that for certain lipids, under certain conditions, simulated in molecular dynamics, the micelle composition does affect the rate of entry, and this gives rise, as written here, such dependence is a prerequisite for homeostatic growth. So we were able to prove that this is not out of the realm of possibility. But most importantly, in the laboratory of Fletcher at Oxford, there have been, in the last few years, a lot of experiments, experimental evidence for homeostatic growth, particularly this particular paper that was published last year. And this shows as follows. you have one head group type, 3 tail group types. So you have 3 different types of lipids. And under conditions of flow, which is non-equilibrium, answering a question that Adi Pross asked, a very good question. This particular experiment is out of equilibrium, away from equilibrium. And what you see is that there is a steady state whereby each of the types of molecules assumes a different concentration. And those concentrations upon a system in which micelles grow and split.
Doron, you got 5 minutes left.
Thank you, I'm almost there.
Okay.
So what happens is that the flow system shows that away from equilibrium, there is compositional preservation, which means homeostatic growth, which supports very strongly our guard model. So I have 2 last slides, and these are the following. First, I asked a lot of questions. So what were the answers? The answers are as follows. This is a summary. Lipids are prebiotically available. Lipids are intrinsically diverse. Lipids assemblies have catalytic properties. Lipids form mutually catalytic networks. Nanoscopic lipid micelles are advantageous for many reasons: size, lability of molecules. Answering the question that Stuart Kaufman is always asked, how will the molecules find each other? Inside a small micelle, they will. That is in a network. Fluid lipid assemblies portray recognition. Lipid micelles can reproduce. Micelles transmit compositional information, and mixed micelles show homeostatic growth. So all of that means lipid first is a highly viable option for early pre-LUCA evolution. And the last slide actually shows the following. Remember that in the non-living chemistries, there was RNA and nanoscopic and lipids, okay? There was a micelle shown there. So now we know that both of them, as shown in the target here, in the title top left, both lipids and RNA harbor catalysis as well as reproduction. This means that both are on equal terms in terms of life's origin. But there is one very strong disadvantage for RNA. If you start from a prebiotic diversity, no matter how it formed, somehow you have to end up with a relatively small number of molecular types that make RNA and protein. What is so wonderful about lipids, the lipid world or lipid first scenario, is that no matter what's in the soup, if there are lipids, namely amphiphils, namely any molecule that has a split behavior with a head that is water-liking and a tail that is water-hating, if those molecules are there, and they are there, then it doesn't matter which they are, and you still get a micelle that reproduces because of the attractor capacities. So the end result is that you don't need to sift out very small number of the most important molecules for life as we know them today. But life can begin with a lot of non-biological molecules and end up along the evolutionary tree, end up by the end of the day with LUCA at the point of at which point we know life is already there. The end of the beginning.